Hey, hi everyone. So today we are going to talk about um, how certain cells from our adaptive side of our immune system um, really help function sort of more like innate effector cells. And this gets us thinking about how our innate and adaptive sides of our immune systems, of course, co-evolved. It's one thing to talk about the innate side and the adaptive side of our immune system when we're learning about it in a classroom environment. In that case, it makes sense. We start off the semester just talking about the innate side, so you can sort of begin to understand how that works, and then we shift over to the adaptive side. But of course, in reality, both sides of our immune system, and I think you've come to appreciate this, now that we're this far into the semester, both sides of our immune system do work together. And so that suggests that along the way, over millions of years of evolution, to have these two sides, there's going to be examples of how certain cells from one side really help out or even function more like the types of cells and types of effector cells and molecules that you find on the other side. So today we'll just kind of take a overview of that and look at some examples. As a result, today, this new material, this is chapter 12, I think, in the book. And so really, I don't go through a lot of the stuff in chapter 12. It's an interesting read, though, if you have the book, because the book talks a lot about the evolution of our immune system and how the two sides were thought to kind of diverge over time. But really, we're just going to spend today's lecture on chapter 12. And as a result, the quiz on this material, the chapter 12 stuff, will be due this Thursday night, as usual. So before I forget, I wanted to say, good job with the exam last week. If any of you are in a situation where you are concerned about where you're at this point in the semester, I invite you to shoot me an email and we can kind of talk in office hours about what's going on to help you finish up the semester, hopefully on a strong note. Okay. All right. So remember, from the very beginning of the semester, we've looked at this cartoon as our way of trying to keep track in our heads of all these different cells that we learn about in this class. We said that the easiest way to think about it is that, well, all of these immune cells that we've been studying, they originate from stem cells in our primary lymphoid tissue, and in particular our red bone marrow, where we have these hematopoietic stem cells that then differentiate, right? Stem cells divide to replenish all of the fully differentiated cells that are used up in your body every single day, right? So things like our white blood cells, our leukocytes, they can typically stick around for a few weeks to a few months, but every day you're going to have billions and billions of them being sort of turned over and recycled. So it's the job of our hematopoietic stem cells in our bone marrow to divide and repopulate all those different types of cells so that we have a nice, strong immune system. So we said that originally, eons ago, multicellular complex living things had innate immune system, right? So the hematopoietic stem cell would have only gone sort of to the right of this cartoon here to develop things like our innate effectors, macrophages, dendritic cells, neutrophils, various granulocytes, etc. But then, around 600 million years ago, when you first had the vertebrates forming, right? The animals that were vertebrates, that's when we saw the development of an adaptive side of our immune system. So you had then the creation of things like natural killer cells. Maybe about 400 million years ago, you saw a divergence. Things like T cells were formed. And then, 400 to 200 million years ago, you had the development of B cells. Right? And certainly when we talk about adaptive effector cells, we think first and foremost of lymphocytes like B cells and T cells. Right? But you do have cells like the natural killer cell that I think ever since the start of the semester, we've kind of conveniently sort of glazed over, right? Because we said natural killer cells sort of represent a development on the adaptive side of our immune system, because, right, they represent the first immune cell that isn't preoccupied with directly attacking and destroying pathogens, right? All the other cells up until then, macrophages, dendritic cells, neutrophils, right? They are preoccupied with just kind of finding something like a bacteria or viruses, destroying them, right? By taking them in through phagocytosis and eliminating them. Even the granulocytes, like things like eosinophils, basophils, and mast cells, right? They're not necessarily targeting a pathogen specifically, but they act in a very generalized, non-specific way, right? When they're triggered, they kind of just blow up all their vacuoles that are carrying these, grains, right? That's what they're called. Granulocytes are filled with all kinds of noxious different types of chemicals like histamines, things like that. And they just release them in a big cloud in a very non sort of targeting kind of way. Right? So natural killer cells then were the first cells to really not care about directly attacking pathogen, but they represented a way to be able to detect, right, cells of our body that had become infected with things like virus. Right? And therefore, natural killer cells would tell the cells of our body that are infected to undergo apoptosis, so that hopefully that would stop the spread of the virus. Right? And then from there, we had more specific types of antigen recognition, the T cells with their T cell receptor, and then of course the B cells with their immunoglobulins, right? So that's why the natural killer cells on this cartoon kind of fall into this funny in-between gray area. So let's explore these natural killer cells a little bit more. So natural killer cells target unhealthy as well as infected cells. So they're very much, you can think of them as like the primitive form of T cells. Both natural killer cells and T cells are cytotoxic. Well, when I say cytotoxic, I mean specifically the CD8 positive T cells are cytotoxic, right? So both these NK cells and CD8 positive T cells work by encountering a target cell, synapsing with it, so their membranes are pressed together, and then hopefully you have certain types of proteins that help them sort of form this nice strong synapse so that other receptors have a chance to kind of see what that target cell has on its surface. Unlike the T cell receptors, though, because remember the T cell receptors, what make them so special is each T cell receptor has a unique, each T cell has a unique T cell receptor on its surface that gives it its own sort of specific antigen detecting capability, right? So different peptides that are being presented on MHC by the target cell that the T cell is synapsing with, it has a chance. The T cell has a chance to kind of check out whether certain peptides match what its T cell receptor is designed to detect, right? So unlike those T cells, then natural killer cells don't have any real specific unique receptors on their surface. They use an array of different types of receptors. And there's a combination of about 30 different types of NK receptors. Each one is good at binding to a different type, general type of molecule that might represent some sort of signal that this target cell, that the NK cell is bunched up against, right? There's something wrong with that cell. Right? So an individual NK cell itself will have only a subset of these 30 different types of NK receptors. Think of it as like a library of 30 different, roughly 30 different NK cell receptor types that you can choose from, and any given NK cell, right, will only express a subset of them. Again, why? So that you actually have the ability for one NK cell to be really good at detecting maybe that handful of different signals. If one NK cell tried to express all of these different receptors, right, then each particular receptor would be limited in the amount that you could have on the surface of the cell, right? And then that means it's kind of like a compromise, right? That NK cell could technically detect 30 different things, but it would detect none of them very well because there's not enough of each kind of receptor on its surface. Therefore, evolutionarily, it makes more sense for a given NK cell to only express a subset of those NK receptors, but then that subset expresses a lot of them. So that particular cell might be really good at detecting, a handful of different signals, even though it might ignore many other types of signals. So when you take into consideration, you have a big population of NK cells in your body, billions and billions of them, then it all kind of evens out, right? Some NK cells would be better for detecting certain problems. Other NK cells would be better for detecting other types of problems. Importantly, these receptors in this library of different NK cell receptors that you can choose from, it's a mix. Some of these receptors are stimulatory or activating receptors. Right? So if the NK cell binds something that this receptor recognizes, it might activate the NK cell, right, and cause it to go ahead and kill that target cell. Other receptors on its surface are inhibitory, right? They kind of discourage the NK cell from killing the target cell. Why? Because, again, I always remind you, think about your immune cells floating around in your body, right? What cells are they typically bumping into? Right? On a given normal day, if you're not really infected with anything, right, they're just going to be bumping into normal, healthy body cells. Right? And so NK cells that bump into normal, healthy body cells, you do not want them to kill that cell. Right? So there must be certain receptors on the surface of the NK cell that act as sort of like the brakes, right? Those are inhibitory, so that the NK cell knows, oh, this is a normal body cell, and then it can just move on, detach, and go to another cell. Here you have an example, this cartoon. So this NK cell, it's showing you that in this particular cartoon, it has 3 different NK cell receptors on its surface. So its whole surface would be covered with lots of these blue ones, lots of these green ones, lots of these orange ones, right? So this particular NK cell is only good at detecting other cells that present these particular 3 signals. So here you have a virus-infected cell. Right, so here's the virus in it. And because the virus cell is infected, many of the proteins that appear on its surface are a little weird looking, right? They've been altered, or there are certain signals that are actually made by the cell in response to being infected. So therefore, part of like the interferon response, right? The virus infected cell begins to display different types of proteins on its surface so that this NK cell that comes along and bumps into it, its subset of receptors on its surface bind, and then maybe this NK cell will be able to detect that this is a virus-infected cell, and the contents of these granules, right, these are these cytotoxic cytokine signals, will then be sent onto this infected cell, telling it to die. So this cartoon on the left is a nice reminder then that you have a big population of different NK cells in your body. Each one of these, so there's 4 of them here in the cartoon, right? This is a nice cartoon because it reminds you that each one of them has a different sort of set of NK cell receptors chosen from the library of 30 plus different ones that could be expressed. And so as a result, different populations of NK cells are better at detecting different types of issues. It should be noted that many of these receptors, right, are common to all NK cells. For example, the CD56, this yellow one that's shown as kind of like the stick here, right? All 4 of these NK cells have it. This CD56 is also present on T cells as well. Right? So in other words, if you're looking under a microscope and you see a bunch of cells, and you say to yourself, I wonder what kind of cells these are, because they just look like cells, right? Maybe you can stain the slide with some antibody that detects CD56, right? And maybe this antibody has some sort of fluorescent molecule attached to it. So then when you go ahead and expose the stained cells on the slide to a particular wavelength of light, maybe you can actually identify, oh, look, those cells are glowing, right? Those are most likely either NK cells or T cells. Right? The trick is you wouldn't be able to tell the difference just by sight, but at least you could say, oh, those are either NK cells or T cells, because both of those types of immune cells have CD56 on them. If you really wanted to see which ones are then NK cells versus T cells, you would then maybe use a different antibody, right? A different antibody that maybe then specifically binds to T cell receptor, so that the T cells would glow a certain color, and you can figure out which ones are NK cells and which ones are T cells. So therefore, the presence of CD56 without the presence of T cell receptor identifies that this is an NK cell, right, when you stain it. So here is just a selection of this big library of 30 or so different NK cell receptors on it. So I don't expect you to memorize this, right? But I show it to you because I want you to really appreciate the fact when I say that a given NK cell expresses a subset of different NK cell receptors on its surface, drawing from a big library of possible genes, right, that the NK cell has in it. It just depends on which subset of these genes actually get expressed to make these different receptors, right? I wanted you to kind of appreciate by looking at this that there's a big selection, right, to choose from. And importantly, that the top versus bottom of this chart divides them into examples of activating NK cell receptors versus inhibitory NK cell receptors. Okay. So let's take a look at some examples of these. Okay. So CD16, right? So CD16 is an activating NK cell receptor. It is also called Fc gamma R3A. Right. So just by that name, that other name, Fc gamma, well, we've heard receptors like this before, right? What does that mean? Oh, yeah. So this must be a receptor protein that binds to the Fc region of some type of immunoglobulin. And since it's Fc gamma, we know, oh, it binds to, right, the stem of IgGs. Right? So the ligand here, the thing that it binds to is an immunoglobulin, right? In particular, IgGs. So let's look at this. Some receptors from this library that are stimulatory, you have to have a situation where the NK cell, when it's pressed up against another cell, needs to at least have a couple different types of these activating receptors signaling in order for the NK cell to activate. That's part of the NK cell being kind of conservative, right? And not wanting to just kind of like freak out and activate against something just because one of its types of receptors on its surface detects something, right? Because otherwise you'd have the NK cell going around in your body killing a lot of perfectly normal, healthy body cells, right? So usually an NK cell needs to see signals from at least a couple of different types of activating receptors on its surface. For example, on the left here, we have an NK cell on the bottom. So these blue things are the granules containing all those cytotoxic messages that would tell a target cell to die, right, to undergo apoptosis. And on the top here, you have a target cell that this NK cell has just kind of bumped into, right, as it's going around in your body. Maybe in this case, this target cell, there is something wrong with it. So as a result, the target cell is showing, these two examples of different types of proteins on its surface that are considered kind of generic signs that there's something wrong with the target cell. One of these is CD48. One of these is this MIC, these yellow things here. So this particular NK cell, from its library of different NK cell receptors, it happens to actually be a cell that expresses a lot of 2B4 on its surface, this orange one, and NKG2D, this one over here. Right? And each one of these is specific for this particular ligand that's presented by the target cell. So as a result, normally you'd have to have these two different receptors activating at the same time. That's what these red arrows are, right? In order to kind of overcome the conservative nature of the NK cell activating. And in this case, this NK cell would activate and maybe tell this target cell to die, right? By releasing the contents of its cytoplasm, of its cytotoxic vesicles over here. However, if the receptor on the surface of the NK cell that activates is this Fc gamma receptor, the one that we said on the previous table binds to IgG, then in this case, all you need is that one type of receptor to bind and activate. Why? Because think about this, right? The fact that you have this IgG floating around, right, this blue immunoglobulin G or this antibody floating around, right, implies that your adaptive immune system is making this antibody as a way of dealing with some kind of a threat, right? So if you have this IgG in your body, either because your adaptive system is making it now because you're trying to beat an infection, or maybe this IgG is present as part of your immune memory, right? Either way, the fact that the IgG is present means, right, at some point, you needed to use this to target and fight some kind of an infection. So that means if you have this IgG and it is attached to the Fc gamma R3 receptor on the NK cell, and this thing goes ahead and binds something. So for example, these little hot dogs over here are the actual antigen, right, that the IgG recognizes. So this target cell, again, might be infected, right? And so it has evidence on its surface of something weird, right? This hot dog over here, right? So the fact that alone, you got the IgG binding to it, that's good enough to convince you, yeah, right? We don't need to see another type of signal here. This NK cell should go ahead and activate and kill this target cell, right? It's a high confidence message, this Fc gamma receptor. All right. So a big commonality that all of our cells have on their surface, of in our body, right? Is remember we learned that all nucleated cells in our body, to some extent, express MHC proteins as a way to display to the immune system, what's going on inside of the cell. Remember, I always say you have macrophages, neutrophils, dendritic cells patrolling around all over all the time, right? They can easily pick up on pathogens that are floating around, right, in the fluids of your body, like the blood or in between cells in your tissues, but they can't look into one of your body's cells and actually see what's going on inside of it, right? That's why it's up to the cells of your body to constantly be presenting small little peptides on MHC platforms on their surface as a way to advertise what's going on inside of them. And so cells like our T cells, right, that are constantly patrolling, they can pick up on whether or not these peptides look weird, look funny, whatever, right? So the very fact that all of your body cells, or most of them, should be expressing reasonable levels of MHC means that the levels of MHC proteins on your body cells is a useful indicator, right, of whether your cell is happy and healthy, right, or if maybe something's dramatically wrong with it, and the levels of MHC proteins might be thrown out of whack, right? They might be either overexpressed or underexpressed. So NK cells, several of the different NK cell receptor types that an NK cell might have, are geared towards just binding to and evaluating, right, how much MHC proteins are on the surface of a cell that the NK cell synapses with. Think about it this way. If you have a cell that's infected, right, by virus, and therefore the normal protein production of that cell is all messed up, or if you have a cell that is transformed into a tumor cell, like on its way to forming cancer, right, its proteins might be all messed up. So there's a very good chance that levels of MHC protein on its surface, right, don't reflect the normal levels that should be there. Either way too much MHC or maybe way too little MHC. Right? Therefore, NK cells use the recognition of self-MHC and detect the levels of how much MHC is there to decide whether to kill a cell. So one of the tricky things about that is that, remember, the MHC platforms are made of these oftentimes very polymorphic HLA proteins. Right? So for example, the human MHC class 1 that presents peptides to things like CD8 positive killer T cells, remember that each one of your cells in your body expresses different variations. There's the HLA A type, B type, C, E, F, and G type. Right? And then the actual shape of your A, B, C, E, F, or G type is dependent upon what version or what allele you inherited based upon your racial, ethnic background, right? Your ancestry. that carries different polymorphisms. Remember, we learned that these HLA genes that code for these HLA proteins are some of the most highly polymorphic alleles in the human genome. And we've been talking about how that's actually good because it allows the human species to have many variations in the population, so that if you have a brand new pathogen that infects the human species, right, some people at least will have certain shapes of HLA proteins that do a better job of presenting things like peptides from that pathogen to their immune system. But the fact that they're polymorphic makes it kind of tricky then, because how can you have a protein on the surface of an NK cell, right, be able to detect MHC proteins on the surface of another target cell that the NK cell is pressed up against if the shape of the MHC is highly variable? Well, not all the HLA proteins are as variable as other ones. Remember the color coding of this cartoon that we saw all the way back in chapter 5 reminded us that the HLA A, B, and C ones, shown in red here, are the most highly polymorphic ones. Right? Other HLA proteins like HLA E, F, and G are more oligomorphic, meaning there's not that many variations, or like the HLA F, right? It's considered monomorphic. There are no alleles that are highly variable in the HLA-F. So therefore, you could have proteins on the surface of the NK cells that detect these, right? The oligomorphic HLA-E, for example. So there's a receptor on the surface of NK cells called CD94 NKG2A, right, that specifically detects levels of HLA-E on target cells that the NK cell synapses with. And that's a good choice because HLA-E isn't highly polymorphic. Right? So we can look at this cartoon here as showing how there's constantly kind of this tug of war between an NK cell binding to a target cell and having different receptors that are stimulatory or activating, kind of competing with other receptors that are more inhibitory, right? And depends on which ones win out. That will influence whether the NK cell leaves the target cell alone, or if the NK cell goes ahead and tells that cell to die, right, to undergo apoptosis. Here we have an example of an NK cell, and it's floating by these perfectly healthy, these are maybe epithelial cells lining, let's say, the respiratory tract or something like that, right? So this is on the tissue side of the of these cells over here. Here, these cells are expressing normal levels of HLA-E on their surface, right? So as a result, this NK cell, it has on its surface here this inhibitory receptor, the CD94 NKG2A that we just looked at on the previous slide. Its job is to detect HLA-Es, right? So remember, the whole surface of this NK cell would be covered with these green inhibitory receptors. So depending upon how much HLA-E is on the surface of this healthy cell, right, that would actually determine how many of these green receptors that detect the HLA-E would actually send inhibitory signals into the nucleus, right? So if you have lots of HLA-E, let's say the normal level, then you'll have lots of these green receptors on the surface of the NK cell sending inhibitory messages into this NK cell. And as a result, the NK cell will say, okay, that's fine, right? I'm not going to activate. I'm just going to detach and float away, right? These other receptors on its surface, like the NKG2D and the 2B4, which we looked at in a previous cartoon, these are activating receptors, right? So this particular NK cell also has these two on its surface. Right, so its surface would also be covered with numerous examples of this receptor and this receptor here. Since this is a healthy cell that it's pressed up against, this healthy blue cell here doesn't express any of the weird, alarming kind of signals that these two activating receptors would bind to. So in other words, right, in this particular case, because it's a healthy cell, the NK cell doesn't receive any stimulatory activating signs but it receives plenty of inhibitory signs, right? So as a result, the NK cell just moves on. But let's say it moves somewhere else in your respiratory tract and it bumps into a patch of infected cells. So here, this is a virus infected cell. And so now on its surface, maybe it's gotten messed up, right? So it's now expressing these weird kind of alarming kind of proteins like the MIC and this CD48, right? And those are the things that are bound up by those stimulatory NK receptors here. So now you're getting plus, activating signals going into the cell, right? But at the same time, maybe the levels of HLA-E on the surface of this virus infected cell have gone down, right? So you don't get much of the inhibitory signal being sent into the NK cell. So as a result, right, the balance shifts. It's a much stronger stimulatory signal. So this NK cell will then activate and it'll release these cytotoxins from its vesicles that will tell this infected cell to die. Right? So you notice the cartoons say inhibits VAV activation or activates VAV, right? So this VAV is a protein that essentially acts as an intermediate protein in the chain of signaling that goes from what's happening on the surface of the cell, through a series of different proteins talking to each other into the nucleus, resulting in the NK cell activating, right? So if VAV is not activated, right, that is essentially what this negative inhibitory signal means here on the left, right? You don't get activation of the NK cell. But if VAV protein is activated, then that VAV goes ahead and it activates various things inside of the NK cell, including the machinery that then begins to unload these cytotoxic signals. All right. So that's great if you have certain receptors that detect these oligomorphic or monomorphic HLA signals. But at the same time, right, all of our cells, nucleated cells, are also expressing those more polymorphic ones like HLA A, B, and C. So it seems like it'd be kind of a waste if your NK cells just ignored the presence of those as sort of useful signs that the cell that it's bumped into is normal and healthy, right? Or maybe there's something weird going on with it. So there are other NK cell receptors that detect these more highly polymorphic HLA types. And those types of receptors we call the KIRs, KIRs. They stand for killer cell immunoglobulin-like receptors. Right. So the way that these KIR receptors work is, I mentioned that some of these HLA genes, and so the shape of the HLA proteins that make up the shape of the MHC platforms on the surface of a target cell are super polymorphic, right? Some of them like A or HLA-B, there's something like 1,500 well-categorized different variations in an amino acid sequence in these proteins. And therefore, it makes it seem like, boy, how can you have an NK cell receptor that can properly detect, right, all those different shapes? The truth is, just because you have maybe 15 different human allelic variations or 1,500 different alleles categorized or cataloged in the human population, it doesn't mean that each one is represented in equal frequency, right? That's an important thing to think about. The truth is the majority of people, right, would have, one type of polymorphism, one allele for that particular gene. Then maybe there'd be a second most common one and a third most common one. And then after that, the frequency of the other 1,000 different variants, right, drops off like really quickly. Right? So the reality is that these NK cell receptors that are in charge of detecting, variations in HLA proteins, so that they can still read whether a target cell has normal levels of these proteins on them, they really only have to be able to detect maybe 3 or 4 different variations in shape. Right? So as a result, for example, look at this here. These pie charts represent the frequency of polymorphisms in these highly polymorphic HLA A, B, and C genes, right, that code for the HLA proteins that make up your MHC1 platforms. And what you see is this, right? Look at HLA-A, right? So there's I think there's something like over 1,000 different cataloged human variations in that gene. But look, the reality is the vast majority of people have one of these 3 different polymorphisms. The gray one here is the majority of the population. And then maybe the second most common one is this BW4 variation. And then maybe this A311 variation, right? The other, I don't know, several hundred, if not thousand variations, right, aren't even popular enough to even be visible as a little pie slice on here, right? Look at HLA-B, same thing. The majority of people have this gray variation, right? Then the second most common one is this BW4, and then this third one, C1, is much smaller, right? And the other 1,000 or so variations, right, aren't even popular enough to be able to show up on this pie chart. When you look at where these genetic variations happen, it's usually, you know, a position in the amino acid sequence of the HLA protein or the MHC platform that does not dramatically alter the shape of the platform. After all, right, the HLA proteins that make the MHC platform, they have to be reasonably uniform and consistent. You can't have like a super weird shaped MHC because then it's not even going to work as an MHC, right? It's not going to hang on to peptide. And even if it did hang on to peptide on the surface to present it on the surface of a cell, right, maybe the T cell receptor of a T cell won't even be able to like recognize it. Right? So most of these allelic variations are very subtle. So if you look here, this is looking straight down at the top of an MHC platform. This is the cleft that holds the peptide, right? So the short peptide that's being presented is this red squiggly thing. And this red dot just shows, oh, this is where one of the genetic variations would be in this position here, right? So therefore, you'd have a particular receptor on the surface of the NK cell that would come and bind to, you know, this region. And you can see this is why they call it the footprint, the shadow of where that particular KIR receptor really kind of cares about binding over here, right? And so you can have slightly different KIR receptors that kind of accommodate the fact that maybe the shape right here is slightly different. Right? Okay, so let's shift gears a little bit and talk a bit about the role of gamma delta T cells as another example of this kind of crossing over between our adaptive side and our innate side. Right? So our NK cells, as we've been talking about for the first part of today's lecture, is a good example, right? Because an NK cell was kind of formed evolutionarily as the start of our adaptive side. But as we've just learned, the way it works is sort of more in an innate kind of way. It just comes, you know, set with a selection of different receptors. Some of them are stimulatory, some of them are inhibitory, right? And then these NK cells will just kind of go around, bumping into different cells of your body. And depending upon that balance of plus minus signals that it gets, that will determine whether that NK cell activates and kills that cell, or if it just floats off to bind another cell. So gamma delta T cells are another example of this kind of crossing over. We've mentioned gamma delta T cells previously because we said when T cells are forming in the thymus, right? Remember when the VDJ recombination starts happening in these precursor cells that came from the bone marrow, they got into the thymus, then the thymus tissue is encouraging the precursor cell, you know, forget about, you know, being an NK cell or whatever, right? You should become a T cell. Right? So then the thymocyte starts doing VDJ recombination. And as we learned, there are a couple of opportunities as the oven mitt, the RAG recombinase protein, is doing the selection of different gene fragments to make things like the different chains that make up a T cell receptor. Right? There's the possibility that it might make a gamma delta T cell receptor. And we said that gamma delta T cell receptor, which is represented, you know, by the little light brown and dark brown version of the T cell receptor, doesn't have to undergo the same tolerance training as an alpha beta T cell. The gamma delta T cell receptor then just heads out of the thymus and goes to work. It does that because those fragments, those gene fragments, the V, the D, and the J that make up the gamma and delta chains are far less varied. Right? There's only a small assortment of these red blocks, blue, green blocks, and yellow blocks to choose from. Right? And as a result, the possible variations in shape of the gamma delta T cell receptor that's made is far more limited than the hundreds and hundreds and millions of possible different shapes of the alpha beta T cell receptor. Again, remember, because the alpha beta T cell receptor, right, you are on purpose making millions and millions of different variations in shape, counting on the fact that some of those shapes will be the right shape to bind to some sort of peptide that's being presented by a novel pathogen, right? And so you're counting on having some T cells, some alpha beta T cells that have the right shape T cell receptors to be able to detect that, so that you can then select that T cell as the basis of your strong adaptive immune response, right? Whereas the gamma delta T cells, right, they don't care about detecting novel peptide sequences because their gamma delta T cell receptors are made from a limited number of shaped possibilities, right? They just evolved that way because these are shapes that are good at picking up on different families of shapes of molecules that are presented by cells in your body that have some sort of issue, right? Either they are infected and so they have a general kind of response showing weird proteins, or these are diseased cells, right, that typically show weird shaped proteins that fit these different categories of shapes that the gamma delta T cell can detect. And therefore, gamma delta T cells don't bind specific antigens. They are more about binding to certain themes or families of molecular signals that represent something being wrong with the target cell that they're synapsing with. For example, there are different metabolites of microbes. So if you do have an infection going on, right, there are many generic types of compounds, molecules that are made by microbes as part of their metabolism that over evolution, right, we see it again and again and again, right? So therefore, gamma delta T cell receptors might be the right shape to bind to those. Certain types of phospholipids that are on the surfaces of cells that are kind of weird looking, right? Gamma delta T cells can bind those. Certain types of glycolipids as well. So these antigens either don't belong in the human body to start with, like the microbial metabolites, or these are molecules that are produced by our body naturally, but normally in very sort of like limited amounts, right? So if you have cells that are overproducing certain of these weird kind of signals, then that's a good sign that that cell, there's something wrong with it, right? And maybe the gamma delta T cell, if it can pick up on that, can tell that cell, hey, maybe you should just undergo apoptosis, right? Because I don't know what's going on with you, but it's not right. So to play it safe, just undergo apoptosis and take yourself out of the picture. Right? So here you can see the gamma chain locus is made of V and J blocks and constant blocks, just like when we studied immunoglobulins and T cell receptor, right? Because after all, this is T cell receptor. But notice, instead of having something like 40 plus different red blocks to choose from, right, the gamma locus here only has like 12 red blocks to choose from, and maybe like, what is this, like 5 yellow blocks to choose from, these J blocks, right? The delta chain, right, has an even more limited number, right? It just has a scant handful of red blocks to choose from, maybe only 3 green D blocks and 4 J blocks, the yellow ones to choose from. So you see what I mean? That when the oven mitt is selecting from these, right, you almost get sort of like only a certain limited number of different shaped gamma delta T cell receptors that can be made. And as a result, you can actually, because there's so few of each kind, you can specifically label them, right? So if we say, for example, this is the gamma chain and these are the red blocks, the variable red blocks here, we can call them V gammas, right? And there's 12 of them. So it'd be a V gamma 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, right? What we found is that depending upon which combinations are put together, like for example, which of these 12 gamma red blocks is chosen, which of these, maybe a handful down here of delta blocks are chosen, right? The resulting shape of that gamma delta T cell receptor, is good for only detecting a particular type of family of weird signals, right? And as a result, gamma delta T cells that are put together in your thymus, right, depending upon what combinations of these different colored blocks are chosen, right, they actually end up being sent to different tissues in your body where there's a better chance of those types of weird signals being the signals that are actually expressed. So for example, if you look at here, gamma delta T cells that end up circulating in the blood, right, typically have combinations of V gamma 9 and V delta 2, meaning the 9th block of the 12 different choices was chosen, and the 2nd block of the delta V blocks were chosen, and that combination makes for the right shape to be more useful in the environment of the blood, right? So the types of three-dimensional shapes it binds to, maybe evolutionarily, right? There's a better chance of those types of shapes being actually expressed by cells in your blood, in your circulation, if they're having problems, right? Other variations are, for example, V gamma 3 and V delta 1, right? Because these gamma delta T cell receptors are more useful in places like the gut epithelium, because that particular combination of V blocks, right, makes for certain shapes that maybe are more suited for binding the types of families of signals that you see in gut epithelium if there's something wrong with your gut epithelial cells. Okay. So this is just a table comparing some of the similarities and differences between the alpha beta T cells that we've been studying in depth when we talk about our adaptive immune system, right? Remember our whole discussion about our adaptive immune response with T cells being cytotoxic T cells or helper T cells, right? And their roles, that has all been focused on alpha beta T cells, right? So today we've taken a closer look at gamma delta T cells, right? This subpopulation of T cells that's made almost as like little detours off of the main alpha beta T cell development in the thymus. Right? I'm not going to spend time right now looking at this, but it's good for you to look this over and just understand, right? I think this is pretty straightforward, and it helps you keep track of the fact that how it is that alpha beta T cells have a very different job than gamma delta T cells. Right? And this is the last slide for today. So you might find that this discussion of gamma delta T cells is kind of curious, right? Because these gamma delta T cells, it sounds like, yeah, these are more like normal maintenance type of cells, right? They just patrol around in your body to different regions of the tissue that their particular gamma delta T cell receptor is maybe better suited for, with the hopes of binding certain families or certain groups of three-dimensional shaped molecules that represent problems in those tissues, right? What is particularly interesting is that these gamma delta T cells, they are important in our bodies, but there's a lot of debate still out there as to how important they really are as part of our normal immune responses. Okay, because certainly the way I describe them to you, it almost seems like they're just sort of like these general things that go out there, and they're not nearly as kind of exciting as the alpha beta T cells, right? But a bit of evidence that really makes things more curious is that these gamma delta T cells are used by different animals with different kind of emphasis on them. For example, in humans, in our bodies, right? If you were to look at this here, this says that of all the blood lymphocytes that are floating through your blood right now, right, T cells, B cells, etc., only 5% of them are gamma delta T cells, right? It's a very small population. Right? And this is the same for something like a mouse. In a mouse's blood, you'll only find 5% of the circulating blood lymphocytes are actually gamma delta T cells. Both in human and mouse, the emphasis seems to be more on the diversity of alpha beta T cells that we can make. Very high diversity, right? And very high diversity in our B cells, immunoglobulin V domains, right? Okay, so you say, okay, yeah, clearly gamma delta T cells are sort of like second fiddle in our bodies and mouses' bodies, right? The bodies of mice. But that's not the same for other animals. Look at in chickens, rabbits, sheep, and cattle. It's kind of surprising, right? Because you might say, well, a chicken is pretty far from a human, whatever. But once you start getting up to other mammals like rabbits, sheep, and cattle, you kind of expect that our adaptive immune systems work more or less the same way, right? But clearly they don't. Because look, in these animals, you have up to 30% of the circulating T cells, lymphocytes in the body of, let's say, a cattle, a cow, or a sheep up to 30% of them are actually these gamma delta T cells, right? So very clearly, other animals depend more heavily on these gamma delta T cells than we do. And interestingly enough, if you look at the diversity in the alpha beta T cells or immunoglobulins in those animals, it's comparatively much lower, right? So there's something about how immune systems work, emphasizing alpha beta T cells and immunoglobulins versus gamma delta T cells, right, that we still haven't unraveled yet. Right? So it's kind of nice because that ends us with today's lecture with this kind of notion that there's still a lot of things about the immune systems of mammals and people more specifically, right, that still remain to be discovered and understood. All right, let's go ahead and stop